Hello my dear students, welcome back to my channel. In the last session, I discussed about translation. I just started with translation in my last session. In this session, I am going to continue. Last session, we discussed what are all the requirements for translation. And in this session, I am going to discuss about the process of translation. But before that, what I want to talk to you, all of you, is if you have not yet subscribed my channel, especially those who are watching for the first time, I request all of you to subscribe my channel. The only intention of mine is to reach all the required students or any other youngsters who are coming to this field. So, I also welcome your suggestions. If you want to give me any suggestions for further improvement also, I always welcome. So this you can mention in my comment box, and I also gave my phone number in my introduction part. That will be given as about. If you open my channel, I think most of you, all of you know it, about. So about me, where I have given about my profile. I have also given my phone number, and definitely you can reach to me and you can ask doubts or any other suggestions also. I welcome, and I also give my phone number here at the end of the here board, so that you can always contact me. You can always contact me for any clarifications or any suggestions or any doubts. You can always message, or you can always talk to me. I always welcome any suggestions. The only intention of starting this channel is to reach the maximum number of students and help them. Because this teaching profession is a passion for me. I have started long ago. I have started my teaching profession, and I am in contact with this field and the students for about a very long time. So it has become a passion for me over the course of time. So I started this channel during this lockdown period, so that I can reach maximum number of students, because most of them are staying at home and attending online classes in various institutions. So I thought, if I start making videos at home, and then I can, to some extent, I can help my students, those who are not able to even reach the online classes, those who are not able to attend the online classes. So thank you very much, children, for listening to me. Now let us come back to the topic. So I'm going to start with translation. Here we will start with process of translation. Process of translation. So in the process of translation, I would like to first list down what are all the steps in translation. So first step in translation is called as activation of amino acids. Activation of amino acids and transfer to tRNA. Transfer to tRNA. So I'm going to list down first, and then I'll go into the details. So activation of amino acids and transfer to tRNA. Second step is called as chain initiation step. Chain initiation step. The third step is called as chain elongation step. Chain elongation. And the fourth step is called as a chain termination step. So in these four different steps, we are going to discuss the process of translation. This I am going to divide in two different sessions, because if I take everything in one session, it will become too lengthy video, and that will be slightly difficult for many to go through that and understand. So first I will complete in one session, I will complete the first step, because the remaining three we have to do at one stretch. Otherwise, you will not be able to follow. So remaining three we are going to do in one video, and this first step, activation of amino acids and its transfer to tRNA, in one video. So first I am going to complete the first step, that is activation of amino acid, and transfer to tRNA. So what is this process? All of you know that for synthesis of proteins, about 20 amino acids are involved. There are 20 amino acids which are required for synthesis of a protein. And these 20 amino acids are present in, cytoplasm. They are present in cytoplasm in the form of amino acid pool. It is called as an amino acid pool which is present in cytoplasm. And here in cytoplasm, these are present in inactive state. In cytoplasm, they are in inactive state. So these amino acids must be activated. Before they take part in synthesis of protein, these amino acids must be activated. And then they are transferred to specific tRNA. Specific tRNA can recognize, one particular tRNA can recognize one particular amino acid. So there are 20 amino acids. So minimum of there are 20 tRNA molecules. But there are 61 codons. 61 codons. So for each codon there is an anticodon. So a specific tRNA is available based on number of codons. So 61 codons. So 61 tRNA molecules are also available. And each tRNA molecule is specific for one particular amino acid. And it can carry only one amino acid at a time. In the previous session, we discussed about the structure of tRNA. We discussed cloverleaf model of tRNA. So that you should go through it, because all these sessions you have to go through it continuously. So let us see what happens here. What is activation of amino acid? So this activation of amino acid takes place in 2 steps. And it is an enzymatic step. What are the requirements here? Here ATP is required, enzyme is required. ATP, that is energy molecule, is required, and also enzyme is required. The enzyme is called as amino acyl synthetase enzyme. Enzyme is called as amino acyl synthetase enzyme. So these are required for the process. So now let us see how the process will take place. Activation of amino acid. So in the activation process, I told you it occurs in 2 steps. So let us do step by step. So first step in activation, first step. So here amino acid combines with ATP, energy molecule, and the enzyme. Enzyme is a specific enzyme. What is the enzyme here? Amino acyl synthetase enzyme. Amino acyl synthetase enzyme. That is the enzyme required. And then it forms a complex called as amino acyl AMP and enzyme complex. The complex is called as amino acyl AMP and enzyme complex, or this is called as amino acyl adenylate complex. Amino acyl adenylate complex. Amino acyl adenylate complex plus pyrophosphate. PP stands for pyrophosphate is released. 2 molecules of phosphate called as pyrophosphate. So when these 2 molecules of phosphate are separated, some amount of energy is liberated. That is required for formation of this complex. Whatever amount of energy released by separation of 2 phosphate molecules, that is required for this step. So here it forms amino acyl adenylate complex. It is an enzyme complex. Now this enzyme complex, it remains in cytoplasm wherever it is taking. Translation is taking place in cytoplasm. So this enzyme complex remains in cytoplasm till it comes in contact with a specific tRNA. Specific tRNA keeps colliding. When it collides with specific tRNA, then it gets charged. It is called as a charging. So in the first step it is called as aminoacyl synthetase. So a complex is formed. Aminoacylation also. This is also called as aminoacylation step. It is also called aminoacylation. Second step is called as a charging. Charging of tRNA. So for charging of tRNA, meaning of charging of tRNA is attachment of specific amino acid to the tRNA. That is called as a charging of tRNA. So in this step what happens? Here what happens is this complex will collide with specific tRNA and first identified by the enzyme recognition end of tRNA. So tRNA structure we have already discussed, and you know the tRNA structure, cloverleaf model. So here we find this is CCA end, that is amino acid attachment end, and this side we have a D loop and this side we have a T loop, and this is anticodon end. This is called as anticodon end. So now what happens here is this complex, amino-acyl adenine complex, will get first attached to D loop because this is an enzyme recognition loop. So it first gets attached to, this is how it gets attached, AMP and amino acid. So this complex now first gets attached to or identified by the D loop. The next step what happens is the amino acid which is formed. The amino acid must be transferred to, this is the amino acid attachment end. So this amino acid must be transferred to amino acid attachment end. So when it transfers to amino acid attachment end, these two will be separated. AMP and the enzyme will be separated. So this we will write in the equation form like we have written the first one. So I am going to write now the second step in the form of equation. Second step we are calling it as charging of tRNA, charged tRNA. tRNA is called as charged tRNA. So when this amino acid gets transferred, we have, I am going to write in equation form amino acid plus AMP and the enzyme. This complex, this is the complex. Now this complex will come in contact with specific tRNA. It comes in contact with specific tRNA and then it forms amino-acyl tRNA. This is called as amino-acyl tRNA. tRNA with amino acid. That is called as an amino-acyl tRNA. Plus the other two will be separated. That is AMP and the enzyme will be separated. AMP and the enzyme will be separated. And this is now called as charged tRNA. It is called as a charged tRNA. Charged tRNA. And in this way, the tRNA can carry the amino acid to the specific site. What is the specific site? It is carrying towards mRNA, towards ribosome, where the actual process of translation will start. And here we can see the first tRNA which is carrying the amino acid. First tRNA is called as Initiator Trna. First tRNA is called as Initiator Trna. This is called Initiator Trna. Later all the other tRNAs will bring specific amino acid depending upon the sequence of codons present on mRNA. Each and every amino acid will get first activated. It first undergo this step and then it gets charged. tRNA will get charged and this charged tRNA will carry the specific amino acid to the ribosome so that it can participate further in translation. With this we complete the first step, children, that is activation step. Here I stop this session. Just the next session I am going to start with the actual process of translation. Because that when we start initiation, elongation and termination, we have to complete in one stretch. Then you will be able to follow. So that's all for now, children. Thank you very much. Have a good day. Please do follow my videos regularly. I can assure you confidently it is going to be a great help to you. Thank you very much.